Hey guys, today I'm going to be talking all about bacteriophages. Here's an image showing a couple different types of bacteriophages. The ones you see on the bottom are kind of the classic bacteriophage with that head and the legs that look a little bit creepy. Those are double-stranded DNA viruses, so they are class 1 in the Baltimore classification scheme. But there are other versions as well, including RNA viruses, single-stranded or double-stranded, and single-stranded DNA viruses. They can have any genetic information that all other viruses could also have, so they're a pretty diverse class of viruses. This is the T4 bacteriophage, kind of is the model for bacteriophages in research. So it's a class 1 virus, and it's a complex virus, meaning that it has a lot of different parts to it. It has a head, a collar, sheath, base plate, tail pins, long fibers. They all kind of work together to inject DNA, which is the T4 bacteriophage's mechanism of entry into host cells. This is a graphic showing penetration, which is the method that these bacteriophages will use to enter the host cell. So first it lands with the tails onto the host cell and then pins. So it has these pins in the underside of the tail region that attach to the host cell, and then it contracts and injects its genetic information into the host. There's one major way to quantify bacteriophages and make a plate with a bacterial lawn, and you'll be able to see little clear regions on the lawn where the virus has infected the bacteria and the bacteria have died. So these little regions are called plaques, and they are counted as plaque forming units because there's theoretically should be made each from one virion that has infected a region of bacteria. They're just like colony forming units, so not all the bacteria are going to form colonies on the plate due to different conditions of plating and other environmental factors. The same goes for plaque forming units. You are going to have a plating efficiency, which is your plaques formed over your total virions. All of the virions that exist in the solution are not going to form plaques. Let's talk about bacteriophage replication. The general mechanism for bacteriophages is lytic mechanism. So they're going to inject their genetic information. It's going to be transcribed by the host cell, or that mRNA from the virus, as it's translated, is going to create proteins which are necessary to build future viruses, and also lyse open the cell so those viruses can spread to other cells in the surrounding region. Whenever the virus first enters a bacteria and the genetic information first enters, you're kind of at this period of the same level of virus while the viral DNA is getting replicated and transcribed and translated. So while that's all happening, you're in the latent period. And this period is also made up of two subcategories: eclipse and maturation. In the eclipse period, you're going to have your formation of new early enzymes. So these are the enzymes that are in your virus that are necessary for the genetic information to actually get through. So those are going to be the ones that are produced first. Then you're also going to get your new nucleic acid production. So your DNA is going to replicate so that these new virions that are leaving will have DNA with them to go ahead and move on to the next cell and also infect those cells. And then within the maturation period of the latent period, you're going to be starting to make your protein coats and all of your proteins necessary for forming the new virion's capsid, or its outer coat, which is completely made of protein. So those are the last things that are formed. And then after that, you're going to start entering the assembly and release period, where your count of virions starts to shoot up at a linear rate. You're making all these virions, and they finally are starting to come together, and you're going to release them to new bacteria. Phage lambda is one important example that we use to talk about bacteriophages. It is unique in a couple of different ways. Firstly, that it has a mechanism of penetration a little bit different from the other classic bacteriophages where it doesn't really have those long tail fibers and still has that tail and those pins where it kind of uses those to penetrate into the host cell. It has a linear DNA inside of the virion, but once it enters the cytoplasm, the DNA kind of circularizes because it has sticky ends. So temperate viruses are viruses that can switch between lytic and lysogenic styles of replication. So this image here shows both of those pathways. The lytic pathway is the basic pathway where the virus enters, the DNA gets replicated, it gets transcribed and translated, and you end up with the new virions that are packaged up. We'll move on to new bacterial cells. However, the lysogenic pathway, the DNA will actually enter into the bacterial DNA and become part of that entire genome, and it will not be transcribed into mRNA. But as these bacteria divide, they'll carry this new genetic information with them into their future generations. So you will end up with a ton of bacteria that have been infected by this DNA. So over time, they can switch into either pathway depending on their conditions. So now you have all these different bacteria that have this viral DNA inside of them, and upon kind of a environmental event or something that causes them to switch, they'll switch into the lytic pathway, and a ton of these bacteria will be infected, and a lot of virions will be produced and escape and cause kind of a widespread infection. The lytic and lysogenic pathways are regulated primarily by the CRO and the CI proteins. As a quick aside, I make the mistake of referring to the C1 protein as CI throughout the video, but it's actually C1. CRO is going to be the major protein that favors lysis, so it activates the lysis genes and inhibits CI, whereas CI is going to favor lysogenesis, so it activates the lysogenic genes and it's going to inhibit CRO. Here's an image of the lambda genome. As we mentioned, it is circular and it has this OR region, which is going to be with CI proteins. They will bind in a certain order onto this region, which causes lysogenic events to occur, whereas the CRO protein will bind in the opposite direction, and that will cause lytic events to occur. So this same region is responsible for both lytic and lysogenic events, but it just responds differently to different proteins. One major factor that can cause the switch between lytic and lysogenic cycles is DNA damage. So when DNA is damaged, Rec-A protease is activated in response to this damage. Protease actually degrades the CI protein. CI is a promoter of lysogeny. So without CI, you're gonna have only the CRO protein, and you're gonna end up activating the lytic cycle. So once you activate the lytic cycle, you're also activating a couple other proteins, oxygenase and integrase. And these are actually going to excise the phage lambda genes from the bacterial genome and allow them to be transcribed. Phage lambda and many other bacteriophages use rolling circle replication. A single strand of DNA can be replicated along the circle and then kind of peel off over to the edge, and you'll form a very long strand of multiple genomes together, which can then be cut and put into one virion and packaged up. We're also going to talk about bacteriophage T4, which is a super important bacteriophage, especially in research of bacteriophages. This is a great way to review the life cycle of a bacteriophage occurs. So bacteriophage T4 is not a temperate virus, meaning that it only enters the lytic cycle. So you're going to have the bacteriophage first injecting and penetrating into the host cell. So you're going to get early proteins, and then middle proteins, and then finally late proteins. You'll have your nucleases, DNA polymerases, and then you're going to end up with your replication of the phage DNA. And then finally, you'll be making kind of the capsid or outer portion of these phages, and then you'll package them all together. And once they're all packaged up, the lysozymes are produced, and that triggers the lysis of these virions. Phage T4 has an interesting way of replicating its DNA. So it has genomes packaged into the virions with slight repeating ends. You can see in the image there's kind of a slight overhang. That's just from the primer as part of the replication, but these overhangs are already present in the virion. From there, recombinase will catalyze the crossing over in these repeated regions to create one long concatemer, which does not have any repeated areas. These ends aren't actually cut off, but they're kind of lost as part of the result of the recombination. The headfuls of DNA are recognized and can be cut and packaged into the phages. However, not necessarily one full genome will be packaged into each phage. The cutoff points can be kind of random. The repeated regions ensure that the entire genome will be transcribed or give a better chance of it in case certain parts of the DNA are interrupted or other things go wrong when the virion is infecting the host. The excess DNA will ensure that it actually ends up infecting the host. Phage T4 also modify their own DNA. The cytosines that are present in the phage T4 genome are hydroxymethylated. So as you can see on the right here, this is a regular cytosine, and on the left, you have your 5-hydroxymethylcytosine. This can then be glycosylated, so adding sugars onto these cytosines. This is a great defense against host endonucleases, which can normally recognize viral DNA and cut them so that they become inactive. That this is a defense mechanism that T4 uses against those endonucleases to make sure that its DNA is preserved. A number of other bacteriophages can also do this, but T4 is one of the main examples. Alrighty, and that is everything. Thank you for tuning in. If you have any questions, make sure to leave them in the comments. Happy studying.